Hello guys, I am here your studyish. I am here with you as reproduction in lower and higher plants. Please do like and subscribe this channel. Reproduction, it is production of young ones like parents. It is essential process related to continuity of species. To maintain continuity of life, organisms produce offsprings showing similar characters. There are two types of reproduction, asexual reproduction and sexual reproduction. Asexual reproduction. In asexual reproduction, fusion of two compatible gametes or sex cells is not involved. Production of genetically identical progeny called clones. In asexual reproduction, progeny from single organism. In this inheritance of genes of parent by progeny, there are modes of asexual reproduction. Modes of asexual reproductions are fragmentation. Fragmentation is multicellular organism. It is in small piece. It is accidental breaking organism. For example, algae, Spirogyra. Budding, it is unicellular organism. It is one or more outgrowth called buds. For example, yeast. Fragmentation in Spirogyra. Budding in yeast. Spore formation, it is different types of formation. It is flagellated motile zoospore. It is zoosporangia. It is biftigellate zoospores. For example, chlamydomonas. Fission, it is binary fission. For example, amoeba, paramecium. Conidia formation, for example, fungus penicillium. Gemule formation, for example, sponges. Spore formation in Rhizobium. Vegetative reproduction, it is a sexual method. Reproduction vegetative plant parts. It is artificial method which is useful in agriculture and horticulture. In vegetative reproduction, new plants and identical to parent plant. Artificial methods in vegetative reproduction, cutting. In this, the small piece of plant part is selected and must possess one or more bud. Stem cutting, example, rose, bougainvillea. Leaf cutting, example, sansevieria. Root cutting, example, blackberry. Grafting, here parts of two plant are joined in such a way that they grow as one plant. In this method, part of stem containing more than one bud, scion, is joined onto a rooted plant called stalk, is called grafting. Whereas budding is also called bud grafting, in which only one bud is joined on the stalk, example apple, pear, rose, etc. Tissue culture, it is a method by which a small amount of plant tissue is carefully grown to given many plantlets. Micropropagation, it is modern method to get plants from tissue culture. Example, orchids. Grafting in rose. Grafting in rose. Sexual reproduction, it involves fusion of two compatible gametes or sex cell, male and female. Organisms reach to the maturity in their life before they can reproduce sexually. It takes place after certain maturity. In higher plants, flowering indicates beginning of reproductive phase. Production of genetically different offsprings. Variations are useful for survival and evolution of species. Flowers, specialized reproductive structure producing haploid gametes. Flowers, specialized reproductive structure producing haploid gametes. Accessory whorls, calyx made up of sepal. Corolla made up of petals. Essential whorls, the male reproductive whorls of flower is called androecium. Individual member of androecium is called stamen. Stamen consists of filament, connective and anther. The female reproductive whorls of flower is called gynoecium. Individual member of gynoecium is called pistil. Pistil consists of style, stigma, and ovary-ovule. Sexual reproduction is two major events. Meiosis is a production of N. In meiosis, male gametes in anther and female gametes in ovule. Fusion of gametes, fertilization, diploid zygote, embryo, new plant, 2N, sporophytes. Structure of anther is mature anther. It is usually diethicous, having two lobes, called tetrasporangiate, having 4 pollen sacs. Monoticous, having single lobe, called bisporangiate, 2 pollen sacs. When young it is homogeneous. Parenchymatous with epidermis. Heterogeneity appears with formation of archisporial cell. T.S of anther, mature anther has anther wall and microsporangium. Anther wall is outer layer. Microsporangium has sporogenous tissue. The wall of mature anther consists of 4 layer, 1. Epidermis, it is outermost protective layer made up of tabular, flattened, cells. 2. Endothecium, it is sub-epidermal layer made up of radially elongated cells with fibrous thickening. 3. Middle layer, it is inner to endothecium made up of thin-walled cells, 1 to 2 layered, which may integrated in anther. 4. Tapetum, innermost. Nutritive layer of anther walls. TS of anther, which consist of epidermis, endothecium, middle layer, tapetum connectives. Dehisced anther, contains pollen grains. Pollen grain, when microspore developed wall around it, then it is known as pollen grain. From archisporial cell, primary parental cell forms anther wall, sporogenous cells forms sporogenous tissue. Microsporogenesis, it is process of formation of microscope by meiosis from MMC called microspore mother cells. Pollen grain is non-modal with single nucleus. Pollen wall and double layer is sporoderm. Pollen wall, exene, outer wall, intine, inner wall. Exene, outer wall, it is thick. It composed of non-biodegradable sporopollenin. It is protective layer. It is smooth or variously sculptured, spiny. It presence of thin areas, germ pores for emergence of pollen tube. Resistance to chemicals. Intine, inner wall, it is thin. It contains cellulose and pectin. It forms pollen tube. It is smooth. Pollen viability, it is ability of pollen grain to germinate and develop male gametophyte. Depends on environment factors, temperature and humidity. It is 30 minutes in rice and wheat, but in some members of family Solanaceae, Rosaceae, Leguminosae, it lasts even for months. Development of male gametophyte, it is considerably reduced. It develops in flowers. Pollen grain, N, is first cell of gametophyte. Pollen grain marks the beginning of male gametophyte. It undergoes first mitotic division to produce bigger, naked vegetative cell small, thin-walled generative cell. The vegetative cell is rich in food and having irregular shape nucleus. The generative cells floats in the cytoplasm of vegetative cells. The second mitotic division is concerned with generative cells only and gives rise to 2 non-modal male gametes. The mitotic division of generative cells takes place either in pollen grain or in the pollen tube. The pollen grain and shed from the anther, at this 2-celled stages most of angiosperm. Structure of an atompous ovule, most common type, chalaza, antipodals, embryo sac, secondary nucleus, synergates, outer integument, inner integument, helium, funicle, placenta, egg, nucellus, micropyle. Ovula are present in ovary. Uniovulate, mango, wheat rice. Multi-ovulate, tomato, lady finger. Parts of ovule, funiculus, is ovule develops inside the ovary and is attached to the placenta by a small stalk called funiculus. Hilum, the point of attachment with the main body of ovule is called helium. Anatropous ovule, curved ovule, where micropyle is near stalk. Nucellus, it is consists of central parenchymatous tissue that forms body of ovule. Integument, the nucelles which is surrounded usually by 2 protective covers. Micropyle, a narrow opening at the apex of ovule is called micropyle. Chalaza, it is base of ovule. Embryo sac, it is over multicellular structure embedded in the nucellus. It is also called as female gametophyte. Female gametophyte is oval, elongated, multicellular 7-celled structured. Megasporogenesis, it is the process of formation of haploid megaspore from megaspore mother cell MMC which is diploid, 2N. Megaspore mother cell is situated in nucellus towards micropelar end. By meiosis, linear tetrad of 4 megaspores. Upper 3 degenerates but lower 1 is functional. Functional megaspore, first cells of female gametophyte. First mitosis, 2 nucleic which migrate to opposite poles. Second mitosis, second nucleic formed at each poles. Third mitosis, 4 nucleic formed at each poles. 1 from each pole moves towards center is called polar nuclei. Pollination, it is transfer of pollen grains from anther to stigma of flower. Pollen grains are non-modal and female gametes are produced at different site. To bring both gametes together. This is necessary act. Agents of pollination also act as agent for seed. Type of pollination, self-pollination and cross-pollination. Self-pollination, it is inbreeding. It occurs in single flowers or 2 flowers in same plant. Autogamy, bisexual flower pollinated by its own pollen shows autogamy. Offsprings produced by self-pollination is genetically identical to parents. For example, p. Cross-pollination, it is outbreeding. It transfer of pollen grains from the anther of one flower to the stigma of another flower of different plants of same species. Xenogamy, two different plants are involved. Need pollination agent. Genetically varied offsprings. For example, food and fiber crops.